Marty.
Hello, Lois.
Hi.
Welcome, Doctor. Thank you for joining us tonight. I'm chair of the program, so and I know we have people who are interested in what you have to say. They've already told me to ask you questions, so we'll see.
Can I ask you a question? Yeah. Am I the only one who's had all the generic? I had 84 tests just done a couple months ago.
Wow.
Is anybody? I had the BRCA and they did 84 testing on it.
84 different genes. Was it an Ashkenazi panel or was it just general carrier testing?
It was probably Ashkenazi, but they did everything through Rocky Mountain Cancer Center.
Great.
Yeah. It still has a question on it, but yes. It still has a gray area that it's had for years. Where'd he go?
The more genetic testing you do, the more you learn about how much we don't know. So.
Yeah.
Marty, you tell me when you want to start.
I was going to say, I think it's Lisa. What do you think? Should we go?
It's 6:32.
Let people trickle in, or are we ready to go?
We can start whatever.
Okay.
I mean, so just so everybody knows, this is being both uploaded on Facebook and then on our YouTube channel. So I hope. Definitely on Facebook.
Okay. Dr. McCandless is with us this evening, and I want to thank him so much during a very stressful time for taking the time to be with us. Shawn McCandless, MD, is a professor of pediatrics and section head of genetics and metabolism at the University of Colorado, Denver School of Medicine and Children's Hospital, Colorado. Boy, I hope you don't have to say all that very often. It's a long one. He's also past president of the Society for Inherited Metabolic Disorders. Dr. McCandless graduated from Temple University School of Medicine in Philadelphia, did his pediatric residency at the University of Wisconsin in Madison, and clinical and biochemical genetics training at Case Western University. He is board certified in pediatrics in 1992, clinical genetics in 1999, and clinical biochemical genetics in 2005. He moved here about 12 years ago in 2018, no less than that. Yeah, way less than that.
three years ago in April.
Okay. If you understood Yiddish, I would say we're landsmen, meaning we have a connection. Doctor worked for the Ohio Department of Health Newborn Screening Advisory Council for the past 12 years. And that was one of my first jobs with the Ohio State Health Department as their clinical audiologist. So we have that little connection, although I was there long before you were. So tonight, Doctor is going to speak on genetics, especially those that influence the Jewish population. So again, thank you so much for being with us.
Thank you for that kind invitation or that kind introduction. And I am sharing my screen. So I hope that you can all see the full-size slides. Is that correct? Great. The topic, the title tonight is a discussion about our current understanding of genetic conditions that are common in the Ashkenazim. And it's a really fascinating topic. And I hope that there's really three parts to the talk. And because I am very long-winded, I typically design my talks so that they can end early because I tend to go on too long. So Lois, you should feel free, or whomever should feel free when we're getting close to the time when I should stop, to just say, let's wrap it up, and I will.
Before you start, let me just say to the participants, if you have questions, please put it in the chat, and then we will read them to Doctor at the end. Thank you.
Great. And as I said, if there's something that I say that's confusing, if you don't hesitate to interrupt, although I'm looking at my slides, so I can't really see the chat box. So if you put something in the chat, maybe Lois can interrupt or someone else can interrupt. the so I actually like to start off this kind of talk, and we already started discussing this a little bit, but just wondering if anybody in the group has had genetic testing or has had experience with genetic disorders, and what that was like for people. So if there's anybody who would like to share or has this, I'd like to hear sort of what people know already about genetics and about genetic disorders in Ashkenazi Jews.
I'll jump in here. And I know Carol Tobin, who just spoke a moment ago about being tested for the BRCA gene, and I remember when I had my kids, the only thing I had ever heard of was Tay-Sachs and a few like Fragile X, what you have there, cystic fibrosis, but there was nothing else that they talked about particularly. And then I heard years later that there are all kinds of related genetic things involving babies particularly. But like Carol, who spoke a minute ago, I had the BRCA test a couple years ago, and I also had it something like 30 years ago. And the number of relationships, these were probably not Jewish relationships, but the relationship between breast cancer and a million other things was unbelievable to me. I think they tested me for one specific thing at that point, and this time around, they had a whole list of about 50 things, which just speaks to me that they've come a long way. I'm sure you'll have plenty to say on that.
And I had a cousin who had, it's not on the list here, dystonia, and she was diagnosed when she was about 10 or 11, and she ended up making it to 13, but she would actually like push, they put her in children's hospital, and she would like put in kind of a semi-adult crib, and she would push her arms on the sides, on the slats of the crib until they broke and things like that. And then there is such a small amount of, apparently it's an Ashkenazic Jewish background on that. And it's so few that there's not even any money to do any research on it.
That's very sad. Was that the familial dysautonomia?
It could be, yeah, because they seem to. Well, no, it came through her. It seems like, yeah, it is familial, and it came out. I'm sorry, yes, it is, because I noticed that her grandmother, kind of, instead of when she walked, it was more like a horse prancing. And then this showed up in this, skip generation type thing. And so I thought that was, and like I said, I was real sorry. You see something like that, there's no money for research, and it's so sad. I'm sorry, go ahead, please, doctor.
Oh, that's very interesting. Somebody else has their hand up. It looks like I can't see.
Hi. I have a question about Lynch syndrome. I don't know if that runs in Jewish families. I know my cousin has that, and she inherited it from her father, unrelated to her mother, so that's good. But it's interesting because my cousin's generation doesn't have any symptoms, but their children have a lot of symptoms. And I was just wondering if you're going to talk a little bit about that.
Sure. Lynch syndrome is one of the inherited cancer predisposition syndromes, often associated with colon cancer, but there's a long list of other cancers that go along with it. And those are interesting conditions, because what you inherit is a change in a gene that increases your risk of developing cancer, but other things have to happen. Just like any cancer is the result of multiple problems occurring in the same cell, where it just sort of loses control and starts to grow and reproduce out of control. That's what cancer is. And Lynch syndrome, like no different than BRCA or other things, is you're starting off with one strike against every cell in your body. And so they're that much closer to just randomly accumulating the additional genetic changes and other changes that allow one cell to grow out of control. And we will come back to talk more about that. I just want to switch over to the slides to say that this first slide is showing disorders that are said to be common to all people of Jewish heritage, whether it's Ashkenazim or Sephardic Jews or Mizrachim. These are seen across all of the various Jewish cultural groups. And this slide basically just shows the carrier frequency for some of these conditions. And all of the ones that are shown here, the carrier frequency is more common in Ashkenazi Jews than it is in people of other ethnicities or other. We often tend to use the word geographic origin because that tends to correlate. Geography tends to correlate with a person's cultural heritage. I would say that the Ashkenazi Jewish people are the exception to that, because as you all know better than I do, the people of Ashkenazi origin have spread throughout the world, but have tended to, for cultural reasons and reasons of faith, of culture, of family, have tended to marry within the culture. And so the genetic contributions have remained relatively stable over many generations within people whose ancestors came from Western Europe, from that part of Western Europe where the Ashkenazim originate. This list, again, I'm going to show you a list that is a complete list of all the disorders that have been said to be more common in the Ashkenazi people than in other groups. And then we're going to talk a little bit about what that means. I do want to just give you a spoiler alert, though. When you start to see these lists of genetic disorders, you begin to think, oh my goodness, we're doomed. How could this be? We're so genetically a mess. But the reality is that genetic disorders, I have not seen any evidence to suggest that genetic disorders are more common in people of Jewish origin than in others. I think the difference is that the mutations tend to be, the mutations that we see in the Ashkenazi Jewish population tend to be the same. So we know more about them than we do about other people. But I don't want you to come away from this talk thinking that somehow your family is at higher risk of genetic disease than other families. There will be, if you look at the long list of specific disorders, your family may be at increased risk relative to the general population just because of your Jewish heritage. But there are, for every disorder that you are at increased risk of, there are probably 25 genetic disorders that you are at reduced risk because of your Jewish heritage and because you have that just tends to be less common. And we'll talk about that in a minute. A couple of things to point out about this list. Cystic fibrosis, which is the third one on the list, that's extremely common. The carrier frequency, 1 out of 26 Ashkenazi Jews is a carrier for cystic fibrosis. That's actually a little bit less common than the carrier frequency for all white people. If you just say Caucasian people or Western people of European American origin or European origin, the carrier frequency is about 1 in 25. So there are several mutations in that gene. It's a large gene. There are more than 1,400 different mutations that have been described. And we'll talk in a little bit what that means, a mutation. But there's at least 1,400 different mutations that can cause this disorder. There's a handful that are relatively abundant in the Jewish population, others that have not been seen in the Jewish population. So all of this is sort of, I think that what we're learning is that part of the issue is that because of the nature of Jewish cultural heritage, that we just know more about what happens in Jewish families than we know in other families. Better studied. people are willing to take part in studies. People are willing and interested and want to learn about their family and about what their, what's going on in their family. And so it's really, I think, important to remember those things, that even though there's a long list of things that if you're of Ashkenazi heritage, there's a long list of things we can say you're more likely to be a carrier of this condition. There's an even longer list that we can say you are less likely to be a carrier of this condition. There is no condition that I'm aware of that has never occurred in an Ashkenazi person. And there's no condition that I'm aware of that only occurs in people of Ashkenazi origin. And we see this, if you look, we talked about those breast cancer mutations, the BRCA1 and 2 mutations that are prevalent in the Jewish population. There's 3 in particular that are relatively prevalent in the Jewish population. Those mutations are also found in non-Jewish populations. Almost always, if you can trace the family history back, someone who's a carrier of one of those mutations now has a Jewish ancestor in the distant past. So the mutations do tend to have originated or be propagated primarily in a Jewish population. There are many other mutations that are found in Finland, that everybody who has that mutation has an ancestor from Finland. Ireland there's some classic variants that are known to occur primarily in people with Irish ancestry. A few things to point out about this list. There are a lot of different types of conditions here. Spinal muscular atrophy, very interesting. It's not an uncommon disorder. It's seen in all populations, but the carrier frequency is a little bit higher in the Jewish population than in other populations. We just started doing newborn screening for spinal muscular atrophy because there are 2 new therapies to treat it, and it's really amazing what's happened in the last 10 years with therapy for these disorders. Bloom syndrome is it's a cancer predisposition syndrome that is also associated with intellectual disability and skin problems. Canavan disease, one of the more common Jewish genetic diseases, is a devastating disorder of metabolism, of a chemical reaction that doesn't occur properly in the brain and causes the brain to deteriorate during early childhood with death in the first 10 years of life. Familial dysautonomia is extremely common in the Ashkenazi population, and that probably right behind Tay-Sachs was one of the ones that sort of led to the development of programs like the Dor Yeshurim and other programs that try to identify carriers even before they get married, to help people make decisions about preventing these disorders, but also just to inform people, because if you know what you're at risk of, you're empowered to figure out what to do about it. So a long list of these disorders. I'm just going to skip to the next slide. This, some of you may have had carrier testing. This, for many if not most of the disorders on this list, if you have 2 copies of every gene. If one of those copies has a mutation on it, but you have one normal copy on the other version, then you will be completely healthy. However, if a person who carries one mutation and is healthy has a baby with someone who carries a mutation in the same gene who's also healthy, and they both pass on the non-working copy of the gene, then the child is affected. We call that autosomal recessive inheritance. Recessive because if you have one normal copy of the gene, you'll be healthy. That's all it takes, is one good one. If you get 2 non-working copies, you'll have the disease. Almost all of these conditions, and that's what this AR here means, autosomal recessive. Almost all of the conditions on this list are inherited in that manner. The testing for carriers for before people got married or before they became pregnant, or even during the pregnancy to determine what conditions a person might be at risk of. That really started in the 1970s and 80s with Tay-Sachs disease in the Jewish population in the New York area. That was where the very first carrier screening programs occurred, and they were wildly successful. Tay-Sachs went from being Tay-Sachs is an inherited disorder where children are perfectly beautiful when they're born, but their brain accumulates a large compound that's found in cell membranes that they can't break down because they're missing an enzyme. Their body is missing a protein that's job is to break down this one chemical. And that is called hexosaminidase. That particular problem over the first somewhere between 2 and 5 years of life, the brain accumulates the storage material, the kid's neurologic function deteriorates, they go blind, and then eventually die. Absolutely devastating. Very common in the Ashkenazim. You can see that 1 in 30 Ashkenazi Jews are carriers of that mutation. Before newborn screen, before the testing program started in New York, it was devastating and it was common. Now, at least in the Jewish community, it's almost unheard of to find a child with Tay-Sachs disease. It used to be when I started in practice, 95% of babies that we saw with Tay-Sachs were of Jewish origin. Today, it's exactly the opposite. Maybe 5% of the babies we see with Tay-Sachs. Fortunately, it's a relatively rare disease, but most of the babies we see now don't have Jewish heritage, or they do, but they don't. The family didn't know it. It was in the distant past, and they've lost that cultural connection, and they were not aware of it. But you can see that 1 in 30 Ashkenazi Jewish people are carriers of that particular condition. If you're just an average American white person, the carrier frequency is about a third less common, 1 in 90. If you're from Thailand, your risk of being a carrier is 1 in 400, 1 in 416. On the other hand, if you're French Canadian from Gaspésie, which I assume that's an area in the Quebec, then you have it's also possible that it's a Native American tribe. I'm actually not sure, but that group has even higher than we see in the Jewish population. And actually French Canadians they're similar to Jewish people. The collection of mutations that are common in that group of people is relatively limited. It's a small number of genes that are abnormal, but they're fairly frequent. They occur frequently. And here you can see that Sephardic and Mizrahim Jews are at much lower risk of Tay-Sachs disease than people of Ashkenazi origin. The point I wanted to make about this was that Tay-Sachs led the way, and the Jewish community in the New York area led the way in carrier screening and using that information to make informed decisions about your life partner, about what you're going to do to become pregnant. And it was wildly successful. As a result of that, as genetic testing improved, we added disorders to the panel of Jewish diseases that we would test carriers for. So when I started off in genetics, we could do Tay-Sachs disease. By the time I came back to Cleveland, Ohio, where I worked for many years, we are with a program that we started. We had the Wolf family of a well-respected family in the Cleveland area had donated a significant sum of money to set up a Tay-Sachs screening program in Cleveland. And about the time we were getting that set up, the ability to sequence more than one gene or to do more rapid, high throughput DNA testing was rapidly evolving because of the Human Genome Project. And by the time we actually started testing, it was actually less expensive to do a panel of about 12 or 14 different genes or mutations that were prevalent in the Jewish population than it was to just test for Tay-Sachs. So we started testing about 14 conditions. That expanded to about 25 or 30 as the program evolved and testing got better and cheaper. And nowadays, it's so inexpensive to do this test that we can look not just for the Jewish mutations, but we can look at the entire gene. We can sequence the entire gene for several hundred genes for less than it cost us to do Tay-Sachs testing 20 years ago, which means that now if you go into your doctor and you say, I want to have Jewish genetic disease carrier screening, they will say, that's not a thing anymore. You can have carrier screening that includes all of the mutations that we know are common in the Jewish population. Plus, we'll look for all the other things that you're at risk of that you might not, that don't have anything to do with you being Jewish. They just have to do with you being human. And anybody can get that testing now. So we don't really break out Jewish genetic testing anymore from just carrier testing for everyone. And what we find is that and that's what this list comes from. This is a program in New York that does this carrier sequencing. And you can see for Tay-Sachs, they're describing how common it is to find carriers in different populations. And they have this for each disorder. And so what you can see here is that for certain disorders, if you look at this tyrosinemia type 1, this is a relatively rare condition, but about 1 in 260 Caucasians are carriers, about twice as common in the Ashkenazi Jewish population. This is a very rare bone disorder that's prevalent in about 1 in 220 Ashkenazi Jews are carrier for. But if you're Caucasian, you're more than twice as likely to be a carrier. So if you're a non-Jewish European American, you're twice as likely to be a carrier for this condition as an Ashkenazi Jew. And you'll notice that we don't have numbers for every population. Now this is, as best I can tell, the complete list of the known disorders that have mutations that are prevalent in the Ashkenazi Jewish population. That doesn't mean that these disorders are all more common in Jews than they are in other people. It just means that there is one or several variants in that gene that are more common in Jewish people than in other people. Just to pick out a couple of examples, Canavan disease, definitely more common in Jewish people than in non-Jews. Gaucher disease, the common form of it that presents in adulthood with bone problems and a large liver and spleen, extremely common in the Jewish population. There are two other, and that's because there are one in particular mutation in that gene that is common in the Ashkenazi people. It causes a very mild form of the disease. There are much more severe mutations that cause more severe disease. We call those Gaucher type 1 or type 2 and type 3. Those are both less common in Jewish people than the type 1. And so even when you look at these individual disorders, you have to be really careful to say that whether they're more or less common in Jewish people. The purpose of this list is, before we could sequence all the genes so easily, we wanted to know what genes do we have specific mutations that we should be looking for in the Jewish population. And this is the list of genes that we know there are prevalent mutations in Ashkenazi Jewish people. Maple syrup urine disease, very unusual, much more common in European Americans of non-Jewish origin. The Amish and Mennonite communities have high rates of that. phenylalanine hydroxylase deficiency, that's PKU. It's no more common in Jewish people than it is in anyone else. There's just a particular mutation that tends to be more common in the Ashkenazim than in others. And so, again, a long list of disorders, most of which you've never heard of, a couple of which I've never heard of, but that for which there has been a mutation identified that is more prevalent in the Ashkenazi population than that particular genetic change is present in other populations. So hopefully that's not too confusing. I do want to show you then the next slide is sort of another list of disorders that are seen primarily in the Spanish and African Jews. And these are some of them. There's a little bit of overlap, but almost all of these are different than the list that's seen in the Ashkenazi people. And we're going to come back in a few minutes to talk about why that is. Why is it that Ashkenazi Jews have one group of mutations that are more prevalent and Sephardic Jews have another group that are more prevalent? It also doesn't, it appears that the prevalence of these things is not as high in the Sephardic and Mizrahi Jews as it is in the ones that tend to be in the Ashkenazim are. And I think that has to do with sort of the heritage and the cultural traditions in these various populations. And I put this one in just to make the point that, as I said earlier, that sometimes being of Ashkenazi origin puts you at higher risk for particular diseases. There are a lot of conditions that being Ashkenazi puts you at lower risk. For instance, Wolman disease. This is a storage disease where the spleen just gets huge because your body can't metabolize cholesterol properly. But it doesn't cause really high cholesterol. It just causes these cholesterol to get stuck in your spleen. And I don't even know if you can read this, but in the Ashkenazi Jews, 1 in 634 are carriers. Worldwide, it's twice that common. 1 in 300 people are carriers. So if you're, so Jewish people are at lower risk of that disorder. Zellweger, the most common form of Zellweger, Jewish people are 5 times or 4.5 times less likely to be a carrier than people than just sort of the general population worldwide. And I bring that up because if you go back, you'll see that Zellweger's on the list. It's on both lists. But it's not just plain old Zellweger. There's a particular form of Zellweger that's a little bit more common in Ashkenazi people than it is in other people. So it's really very much about the specific mutations, not about a people being at higher risk of genetic disorder. And we're gonna talk about why that is. And to have that discussion,
Dr.
we need to.
Someone asked about Down syndrome. Is it higher in the Jewish population?
Great question. The answer to that is no. And that's because Down syndrome comes up new in the egg or the sperm that baby comes from. And there are reasons why that happens. It's, as you probably know, Down syndrome is usually in the formation of the egg, rarely in the formation of the sperm. When you start off with 2 copies of every set of genes, when you're making eggs and sperm, you divide them into 2. So you have 1 copy of the genome in the egg and 1 copy in the sperm. And when a sperm and an egg get together, you make a brand new person with a whole unique set of genes, 2 copies of each. If that chromosome 21 doesn't separate so that you make an egg that has 2 copies, then when the sperm fertilizes that egg, you end up with 3 copies of chromosome 21. And it just so happens that chromosome 21 is the very smallest chromosome, has the fewest genes on it. And so it's the one that you're most likely to be able to survive if you have too much of it. So Down syndrome is associated with, as women get older, more of their eggs have those kinds of abnormalities in them, which is that's an interesting topic for another day, because a woman's eggs are all made before she's ever born. those, you're not, men are sperm factories. They're making sperm their entire life. And men, as they get older, accumulate mutations like the ones we're talking about this evening. Women, on the other hand, their DNA is copied, they make their eggs, and they're just sitting there in storage until they need them. We don't know for sure why, but as women get older, they tend to produce more, they release more of the eggs that have genetic errors in them. In particular, that error where you have two copies of a chromosome instead of one. And it can happen with all the other chromosomes, but most of them are not compatible with life. And so the babies, they cause an early miscarriage. Now, there are some characteristics of Ashkenazi Jews in the United States and in Europe that might increase the prevalence of Down syndrome slightly. And that is that more highly educated people tend to have their children later in life. And the older a mother is when she has her babies, the higher the risk of having Down syndrome, with the exception of the very early teenage years. So the risk of Down syndrome is highest in 13-year-olds and 40-year-olds mothers. And so because traditionally, traditionally Jewish populations have tended to value education a little bit more highly than some other populations, there is a tendency for Jewish women to be a bit older at the time of marriage, and that could lead to a slightly increased risk of Down syndrome. It has nothing to do with being Jewish. It has nothing to do with your genetic background. It has to do with your choices about when to start your family. Great question. Thank you. So as we getting back then to the specific mutations in genes, just kind of thinking about how is it that one mutation is more prevalent in one group of people than in another. And it helps to think a little bit about how we know how genes have moved through the population. And there's a couple of things that are really helpful to know. First off is that the chromosomes, and I probably should have, we'll talk about this more later, I should have probably put this in earlier. Chromosomes are just long strands of DNA. And each strand of DNA has multiple sort of instructions for how to make a protein. And we call that a gene. So a piece of DNA that's the code for how to make a protein, that's a gene, essentially. It's a little bit more complicated than that, but for now, that'll be a good explanation. A single strand of DNA can have thousands of genes on it. They're each, as you go down the chain, it's a different gene, a different instruction. And we each have, there are 23 chromosomes, and we each have 2 copies of each chromosome, so that we have a total of 46 chromosomes, 23 that came from our mother, 23 that came from our father. The thing that makes us male and female is there's one particular chromosome called the X chromosome that has, instead of having an identical partner, there's a much smaller variant called the Y chromosome, and they have different genes on them. So if you have 2 X chromosomes, that's a girl. You'll be a genetic female. If you get an X and a Y, you'll be a male. So it follows, therefore, that fathers can pass on an X or a Y chromosome, but mothers can only pass on an X chromosome. So the Y chromosome is always inherited from the father. A boy inherits it from his father. So we can follow changes across the world in the Y chromosome, and it tells us about men. Now, that's what we call the genomic DNA. That's the DNA that's in the nucleus of the cell. You have identical copy in every cell of your body. You have one set of chromosomes in every cell of your body. Inside every cell of your body, there's something we call an organelle that's called the mitochondria. And you might have heard of the mitochondria. The mitochondria is a little membrane-bound component of a cell. And the main job of the mitochondria is to convert the chemicals in the food you eat, to break those chemical bonds and convert them to energy. The mitochondria, we think in evolution, came from one cell absorbed a bacteria, or probably an archaea type of bacteria. And that bacteria, rather than just sort of being destroyed in the other cell, it just kind of survived in there, and it made energy that the other cell could use. And that symbiotic relationship worked so well that bacteria that had gotten engulfed in the other cell through evolution just became part of the cell. So the mitochondria have their own little tiny piece of DNA. It's just a little tiny piece of DNA. Instead of it only codes for 13 proteins, whereas the nuclear DNA codes for 20 some thousand different proteins. But each mitochondria has its own little piece of DNA that makes a few of the proteins that your mitochondria use for that energy metabolism that we talked about. It turns out you have more than one copy of that mitochondrial DNA in each mitochondria, and each mitochondria, you might have a couple of hundred mitochondria in each cell. So we have probably we have 1,000 of more copies of the mitochondrial DNA than the genetic DNA. Mitochondria are passed from generation to generation only from the egg. So they only come from the mother. So if you look at changes in the mitochondrial DNA, you can trace a person's maternal lineage back for as far back as you want to go. That's why when you hear about that book, The Daughters of Eve, that's about DNA, they're talking about the mitochondrial DNA. And you can look at mitochondrial DNA and you can say, if you have this sequence, your mother's family came from this part of the world or came from that part of the world. The same can be said of the Y chromosome for men. So we can track mothers and we can track fathers separately through history. The other thing that is important, the other concept that's really important to know is that when there's something called a genetic bottleneck. And the best example that I'm aware of this is the Amish community. So you have seen Amish Dutch community. There's a couple of communities in Colorado where Lois and I both spent time in Ohio and Pennsylvania. There's large communities. There were 2 shiploads of religious dissenters who came from Switzerland in the early 19th century, late 18th, early 19th century. 2 boatloads, each had a couple of hundred people. All of the Amish and Mennonite people in the United States today came from those 400 or 500 people. So if you think about it, think of all the genetic diversity that was in Europe at that time. Every gene had variants that were all over the place, but you took 400 of those people and you moved them to North America. What you did was, that's called a genetic bottleneck. Whatever mutations those people had, those are the mutations that you're going to see in all of their ancestors, everyone who comes from them. And so that rich pattern of rare variants that was present in Europe 200 years ago was squeezed down to just a few hundred variants that were present in those people that came over. And all of the Amish and Mennonite people alive today that's where their genetic variation came from. And so there's a whole bunch of genetic changes that are prevalent in that population because of that genetic bottleneck. That group expanded from a small number of people. The exact same thing happened with the Ashkenazi people, except that it was 800 or 900 years ago. A very small number of individuals in Western Europe, possibly as few as 300 or 400, intermarried, expanded, grew, traveled around, but continued to intermarry, so that whatever mutations were prevalent, happened to be in those 300 or 400 people, those are the mutations we see in Ashkenazi people today, among the hundreds of thousands of Ashkenazi people today. That's where they came from. And that's why the list is actually so narrow. It's really the diversity of common variants that we see in the Ashkenazi people today are a reflection of the sort of general genetic variation that was present 900 years ago in Western Europe. It's like you took a small sample and then you expanded that sample. And it's really interesting because, as I point out here, that the timing of sort of the Ashkenazi people spread through the Holy Roman Empire. And because these conditions are almost all recessive, it's very unusual that a parent, even though you're more likely to be a carrier, it's still uncommon to have an affected baby. And so it didn't really reach attention. Nobody sort of said, hey, we've got a problem here, because they're rare diseases. Even in the Ashkenazi population, they're rare. There have been several other genetic bottlenecks over time, and any place where you have a community where you tend to interbreed. So Jews in New York, Jews on the east side of Cleveland, maybe Jews in Colorado, I don't know, who tend to marry each other. You probably have little genetic bottlenecks that have happened in each of those areas. That's true of any population. I grew up in Western Pennsylvania, where a lot of Scots Irish people immigrated to 250 years ago. And they all, they lived in the hills of Pennsylvania. It wasn't easy to travel around. They all married in that small group of people. And so there's a group of disorders that are more common there than they are in other places. I'm going to skip this. I don't think you may not care too much about the history of DNA and genetics and how we learned all this. Some of us think it's really interesting, but most people I talk to don't. I want to talk a little bit in the last few minutes about just human complexity. What makes humans so complex? And, what humans like to feel that we're really special, but the more that we learn about ourselves, the more that we learn that we're actually not all that special. And so, when you look at the complexity of our brain and the complexity of who we are, it turns out that we are more complex than a lot of other organisms. So why is that? Is it because we have more DNA? Well, the answer is no. Homo sapiens, that's humans. We have 3 billion bits of information in our genome. This lily that grows in the mountains in Japan has the largest known genome. It's about 50 times larger than the human genome. That plant, every cell in that plant has 50 times as much DNA in it as a human cell does. 150 billion bits of information. And yet, that flower doesn't have a brain. That flower can't walk, can't write a novel, can't produce gene therapy or the cure for cancer. Now, that's not entirely true. Some plants actually have developed cures for cancer, but not on purpose. But it's certainly not the size of the genome that determines our complexity. So then the question is, well, maybe it's not how much DNA you have, but maybe it's how many genes you have. So humans have maybe 23,000 genes, 23,000 instructions for how to make a protein. We used to think, before the Human Genome Project, we thought that number was about 100,000 because we had identified about 100,000 different proteins in the human body. But we now realize that many of those proteins are just variants that come from the same gene, and that we really only have about 22,000 genes. Even when I made this slide a few years ago, we've gotten smarter since then. So that's still a lot of genes, but it's nowhere near. The corn plant has 40,000 genes. This little water flea that I'm showing you this microscopic picture of has 31,000 genes. So it has 50% more genes almost than humans, 40% more genes than humans, but certainly not as complex, doesn't have a brain like humans have. So where does this complexity, what makes humans so special? And what makes us special is that we probably have the most complicated, what we call regulation of genes. We have the most complicated system for turning genes on and off at the right time and the right place, and making just the right amount of whatever the product of that gene is. We also, humans and other animals, have evolved ways of changing proteins after they're made that can alter their function. And we call that post-translational modifications. We found ways we found. We didn't really find them. They just evolved by natural selection. But there are individual genes can be spliced together in different ways to make different kinds of proteins. And that's pretty cool. And so there's this long list of characteristics of how genes get turned on and off in the right time, in the right place, and then what happens to the products of those genes after they're made. What do we do to them after they're made? That creates all these novel functions that make us these wonderfully complex beings that we are. And the genes themselves are a very modest part of that. That said, if there's a defect in a gene, it tends to have profound effects. So much so that most genetic changes, we don't even, you can't even survive to be born with them. I'm not going to really talk much about the tools that we use. I wanted to mention that because of the Human Genome Project, which started in 1993 and finished up around 2003, one of the results of that was that we now know the sequence of the entire human genome. And I say that knowing that there's actually still a few little areas that are really hard to sequence, that we're not 100% sure of what the normal sequence is. But because of that, as part of the Human Genome Project, it led to really rapid advances in the technology for sequencing DNA. So when I started off in genetics, a person could get a PhD for sequencing a gene. You could spend 5 years sequencing a gene, one copy of one gene, and get a PhD for doing that. Today, you can come to my clinic. I can take a swab from your cheek and send it to a lab with a check for $1,000, and they will send us back your entire genome, every nucleotide, in 3 or 4 weeks. The first human genome that the Human Genome Project came up with cost, I want to say it was on the order of $100 billion. Today, you can get that genome for a couple of $100. And in fact, it's so cheap to get it. The real cost is not in doing the sequencing. The real cost is in for somebody to actually sit down at a computer and look at the data and figure out whether it's normal or not. The other thing we've learned from that, and now that we've actually sequenced thousands of individual genomes, actually probably close to 1 million have been sequenced now, we've realized that we don't really know what normal means because everybody's different. If you were to sequence the genome of every person that's listening to this talk, we would each probably have somewhere between 15 and 20,000 variants that had never been described before among our 3 billion bits of genetic information. And so that really is humbling to a geneticist. It makes us realize that the more we learn, the more we realize how much we don't know. And it's an exciting time to be a geneticist. It's also exciting because knowing what's in the human genome, and just even to be at the level now where we're beginning to understand the complexity of the genome and know most, if not all, of the genes, we're also learning a lot about how genes affect health and how they affect disease. And we're better understanding how different chemicals that we refer to as drugs interact with genes and the products of their genes in a way that we're much better able to develop a new therapy today than we were 25 or 30 years ago. In the past, if you wanted to create an antibiotic, you would take some bacteria in your lab and you would just add 3,000 different chemicals one at a time to see if anything killed the bacteria. And if you found something that killed the bacteria, you would then give it to some animals to see if it killed the animals or whether the animals could survive. And if you found a chemical that would kill a bacteria but let an animal live, that might be an antibiotic that you could use. It was a very hit or miss sort of trial and error process. Today, if you want to develop an antibiotic, you will do a genetic sequence of that bacteria. You'll determine the structure of all the proteins in that bacteria, and then you'll design a chemical that will interfere with one of those proteins. It will be designed from beginning to end, including the process to produce the drug. The only part that's still trial and error is we still give those new drugs to animals to make sure it doesn't cause harm, unexpected harm, because we're not smart enough to figure out everything just by the genetic sequence and looking at the proteins. So it's really changed medicine. And if you follow medicine, if you follow the pharmaceutical industry, you'll know this, that when I went to medical school 30 some years ago, there were a list of drugs that were very commonly used. The list of drugs available to us today is about 5 times larger, and very few of the drugs that we used back then are still commonly used today. And the drugs we have today are much better. And it's all because of this additional knowledge we have of the genome. And in large part, that was driven, for what it's worth, by a lot of Ashkenazi Jews who did this research over the last 100 years, and by what we've learned from studying the genetic variation that's common in the Jewish people. And so we've kind of gone full circle then. We recognized there were disorders that were more common in Jews than non-Jews. We learned a lot about that because that group of people was willing to learn and wanted to know more about themselves. That led, along with other changes in technology, to rapid advances in testing that now mean that we can test anyone who wants it for hundreds of different to find out they're carriers of hundreds of different conditions. And it's really quite economical. And so when I look at the people on this call, I think most of us are probably not going to be having babies. I think we're done with that, most of us. I don't want to speak for all of you, but I think most of us are done with that. But we have children, we have grandchildren. And I would encourage you to talk with them about carrier testing well before they're ready to start their family, just to find out what they're at risk of. So that they can go into their pregnancies with good information. Just today, I'm taking care of a baby in the hospital that we diagnosed prenatally because mom had had carrier testing. And we actually started the therapy for this baby before the baby was delivered. We gave the mom the treatment for this. This is a condition that typically presents at about 4 days of life with baby in a coma because of ammonia accumulating in their blood. And this baby has now been treated since before birth and is spectacular, is just thriving, doing everything a normal baby should do, and is not going to have a coma because of high ammonia, because we knew what to look for and we knew how to treat it. So when my daughter was carrying her first baby a couple of years ago, she said, my obstetrician's offered me this testing. I'm not really that interested in it. I said, oh yeah, you need to have that testing. And she did. And we got really good news that she and her husband are not carriers for the same things. But I encourage my children to have this testing, and I encourage you to encourage your children to have it too, and your grandchildren. I think this is probably a good place to stop, Lois, if you agree with that.
We have 2 comments. One is to tell you that, let's see, I can find it now. That in 1977, here it is, Tay-Sachs was well known in Denver. There was a family with 2 adult children that had it. And at that time, she believes only 8 adults in the world had diagnosed with the disease. And then when I was doing audiology at a high-risk Denver preschool in 1988, I walked in and the teachers tell me that this one little kid was Fragile X. And I went home and I said to my husband, who used to be a pediatrician, what is Fragile X? And he looked at me, he says, I've never heard of it. By then he was in radiology. So that's, how new things are. Then a number of years later, I get a call from my cousin whose first child had Cruzon's disease and died at like 9-10 months of age. And of course, then she had testing before her second pregnancy. They didn't know about Fragile X. And he has Fragile X. He's still alive. Luckily, it's on the other side of the family. It's on the father's side, and I'm related on the mother's side. But very debilitating. And it's so sad because he or she went through the testing and it just wasn't known. I think that would have been 1976 that he was born, because he's my youngest son's age.
Very interesting, because Fragile X syndrome, I'm sorry, I interrupted you, Lois.
What you do today is just absolutely wonderful and change people's lives.
It's fabulous. And Fragile X, I will say, is in large part because of people at University of Colorado who did much of the seminal work in understanding that condition, understanding how it's inherited. Fragile X is really interesting. It's on the X chromosome, so it's always inherited from the mother. Although a father who is a carrier can pass on the carrier state to his daughters. But the mutation in Fragile X is one we call it a triplet repeat expansion. So if you look at the gene, it's got this little stretch where it goes, there's 4 building blocks of DNA, A, C, G, and T. And those are the first letters of the chemicals that make them up, adenosine, cytosine, and so on. If you look at the Fragile X gene, there's a stretch where it goes CCG, 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 up to 20, 25 times is normal. Well, you can imagine when you're copying that, every once in a while, the machinery will slip a little bit and you'll add some extra copies. And the more copies you have, the more likely you are to slip and add even more copies. And so over generations, that genetic change can get bigger and bigger. And once it gets to about 200 copies, it causes a severe problem with development of the brain. It's actually one of the most common inherited forms of intellectual disability and autism that we see. The interesting thing about it is that you have to do a special genetic test to find those kinds of mutations, because they don't show up in a regular DNA sequencing that's just looking what letter is here. And that's because those parts of the genome where there's repeated sections like that are really hard for our machines to sequence. We're not very good at sequencing those. So you have to do special testing for Fragile X. So if you were to send your DNA to one of these labs that does that testing, they would do sequencing for most of the things I showed you, but they would do a separate DNA test specifically for Fragile X syndrome. It's also really interesting in that the fact that it expands when it's passed from a mother to her children. It can also get smaller, but we don't really know when it gets smaller. We only know when it gets bigger because a healthy person has a child who's affected. We know that males and females who carry the what we call pre-mutations, more than the normal amount of repeats, but not enough to cause intellectual disability, those people later in life, the carriers, can develop tremor and a little bit of difficulty with balance. It's called Fragile X tremor ataxia syndrome.
Are there any other questions?
Oh, yeah. There's one on the screen here, too. The question, do you have it, Lois?
I see it, about is there a known genetic mutation that causes autism?
Right.
There are probably at least 150 different genetic changes that we know that cause autism, but that accounts for probably less than 15% of all cases of autism, and the other 85% we don't have a genetic test for today. And many of the known genetic causes of autism have other findings along with it that makes it apparent that it's a part of a syndrome. So we call that syndromic autism. And I would say Fragile X is one of those. So if you see a child that has autism, who has the typical physical characteristics of Fragile X syndrome, then you would do Fragile X testing in that person. But run-of-the-mill, not that there is any such thing as run-of-the-mill autism, but sort of the typical autism, we think almost certainly there's a genetic basis for it. We don't know, but we don't know what that is in most cases. We don't know whether it's a single genetic change or whether it's a combination. We think it might actually be the combination of multiple genes interacting with each other, because if you have two parents, both of whom have autism in their family, even though they don't have autism, they're at higher risk of having a child with autism than two parents who don't have people with autism in their family. And we think that there might be a situation we call multigenic inheritance, or multifactorial inheritance, or sometimes people talk about polygenic inheritance, where maybe there's 100 different variants of genes that make you more, you've got 100 different genes, and a variant in each one of them can make you more or less likely to have autism by just a little bit. And you put them all together, and some of them increase your risk, some of them increase your risk. And the balance at the end of the day is most people are kind of in the middle and don't have autism. But some people will just by chance get a combination of those genes that puts them at higher risk of having autism. And if because of they have a sort of more of the variants that make you prone to autism. And if two parents that carry that excess burden get together, they're more likely to have a child with autism. That actually reminds me of something else I think is a little bit interesting. I'm not sure this is true. And in fact, I'm pretty skeptical of it, but I think it's worth mentioning because you might be interested in it. Some people have said, why these genetic changes in the Ashkenazi people? Why are these the things that we see? And some people have speculated that there's also some data to suggest that if you do IQ testing in people of Ashkenazi origin, the average is a little bit higher than it is in the general population. And so some people have said maybe there's a selective advantage to all these autosomal recessive conditions, because many of them are involved in brain development or affect the brain. And maybe if you have these variants, maybe that somehow makes your brain work better. But if you get two copies of it, it really is quite damaging. I'm not sure I believe that, but I think it's a really interesting, it's an interesting way to think about it. It's be really hard to prove.
I have a question or two, if that's okay. I
Sure.
know that there's a couple things I wonder. For instance, if with Alzheimer's, how far are we in figuring out? I know about the tangles, and I know about some of the latest research beyond that, but are there things do you believe that there are things we can do to prevent it in ourselves or other people? Are we just destined to get it or not? I know they're studying all of this, but genetically, do you think there's some inherited tendency towards it, which I've often heard?
Yeah, there certainly is. There's definitely a there are inherited factors that increase a person's risk of Alzheimer's. There are a few extremely rare causes of Alzheimer's that if you have the genetic change, you will have Alzheimer's. The hope is that even those really rare causes of Alzheimer's will eventually be able to treat, because you don't have it when you're young. And so if it's something that's progressive and changes over time, you you would think that if we could figure out the problem, we might be able to treat it or fix it. There are other factors that are risk factors for Alzheimer's. The most famous one is there's what's called an apolipoprotein. Those are proteins that are involved in carrying fats and cholesterol around in your blood. And there's one form of that called ApoE, that there's 4 different variants of ApoE that you can have. And one of them, if you have 2 copies of it, you have an increased risk of Alzheimer's. It's not 100%, but it definitely increases your risk. We don't really understand why. There are lots of other genes that we know there are variants in that increase your risk for Alzheimer's. The more, and again, I'm not an expert in Alzheimer's, but the more we understand the pathways that are involved by identifying those genetic changes, we can then start to see how the genetic changes are interfering with the way that sort of pathway of works, and then we can start to intervene to reduce the risk for those people at higher risk. Right now, though, I'm not aware that anybody's recommending testing for risk for Alzheimer's.
Well, because it can't solve anything. So if you know you're going to get it, who cares, I guess.
Right. Yeah.
I mean,
right.
I guess the second thing I would ask is someone asked me to ask about this. Someone I know, her daughter's having a baby, and the mother was just diagnosed with CLL leukemia.
Oh.
Is something like that brought on by anything, or that's just chance? Same with any cancer that your child inherits, or not inherits. Do your children and your grandchildren? Should we all be worried about that factor?
Yeah, there are lots of different genetic changes that predispose someone to cancer, and I can't prove this, but I've always felt that the earlier a cancer starts, the more likely you are to have a genetic predisposition to that cancer, because most people live their whole life and don't get cancer. Most people that do get cancer get it when they're in their 80s or late 70s. So and this is certainly borne out with breast cancer. If you get breast cancer when you're 80 years old, there's the likelihood that you're going to be a carrier of a BRCA mutation is low. If you get breast cancer when you're 35 years old, the probability that you have a genetic change in BRCA or another gene that predisposes to breast cancer is very, very high. I extrapolate that to childhood cancers. I think that cancers that occur in children, it feels to me like there has to be a genetic predisposition. We don't understand what it is, though. We don't have a clue for most of them, but we're working on that. There are lots of labs working on that. CLL in particular, I don't know off the top of my head about genetic predisposition to that. Most of the blood-borne cancers, though, don't tend to run in families. They tend to be sort of one-off things. And it may be that when somebody gets that at a young age, that they have a genetic change that came up new in them that set them off on that path at an earlier age. And for most cancers, we think that you get one mutation in a cell, and then another mutation occurs in that cell, and that you accumulate mutations until you get one cell down the line that has 6 or 8 different mutations that all work together to make that cell lose the control over the way it grows and divides. And that's what the cancer is. So if you start off with one mutation, then you're already partway down that pathway to developing cancer, and you're likely to get cancer at an earlier age.
And as far as other diseases, I'll make this quick because there are people asking questions, and I do want to let you go, which would be nice, wouldn't it, after you've talked for over an hour? Are there still, for instance, this is off the wall, both of my husband's parents had polycythemia and died of it. There's been no sign of it in any of the other generations. So was that, and they still don't know to this day, I don't think. So is that kind of thing or where you find some weird thing in your kid, is that just a one-off, like you just said about the, I asked before?
Polycythemia is a great example because that seems to be an acquired mutation in a gene, I think it's called JAK2 or JAK22. That causes that. And it's thought to be, it's just a random thing. It's not generally inherited. At least the families that I've investigated, it was not inherited. And from the last time I read about it, it was not thought to be typically inherited. So there's, and that's the other thing is that we have a much better understanding of the childhood genetic diseases because they're more obvious. You have a change in a gene, that gene is responsible for causing this condition. Needless to say, in adults, the problems are more complicated. there's usually an interaction between environmental exposures and all the bad things we do to our bodies over the years and our genetic predisposition that work together to cause disease in adults. I'm a geneticist, so I think everything's genetic, in my opinion. So it's that we're all born with risk. We're all born with predisposition to disease, and if we could better understand those, that's where I think the promise of precision medicine is. If we could understand when we're young, what are the highest risks for us? What do we need to avoid? What do we need to do to prevent us from getting cancer, from getting other things? I think that would be really the dream for precision medicine and for genetics in general. Now, I see that Mr. Tobin asked about Gilbert disease, and that is hereditary. It's a autosomal recessive condition, so you inherit the gene from both parents, and it's basically in a gene that helps break down bilirubin in your liver, and so it's people that have Gilbert. It's a totally benign condition as far as we know, but people that have Gilbert just always have a high bilirubin, and they always have this kind of bronze yellowish coloration to their skin.
I don't have that. I don't have the bronze coloration, but I do have the disease.
You do have the disease. Well, if you have to have a genetic disease, that's a good one to have because it generally doesn't cause problems, I think.
Well,
I hope that's true for you. They what?
You tell me not to have so much alcohol because it can affect it works on the liver a little bit.
Well, there is that. I'm of Irish heritage, and we have a condition that's called hereditary hemochromatosis. It's common. That's actually common in lots of populations, but that also predisposes. If you've ever known somebody who developed cirrhosis of the liver when they were 50 or 60 years old, and their doctor said, you must be an alcoholic, and they said, I'm not an alcoholic, they probably have hereditary hemochromatosis because that's a well-known association.
Just a little bit of an aside. In the early 60s, they took genetics, and they were still making references to Mendel and the Drosophila. And what you're doing today is so fascinating. It is unbelievable.
It is fascinating, but it all builds on that. You know, Mendel and the people that came after him, that you know, Watson and Crick and Rosalind Franklin and Morris Wilkins, who figured out the structure of DNA. That helped us better understand. Thomas Hunt Morgan, who had the famous Drosophila lab, I think it was at NYU, and we still use Drosophila. They're a very powerful tool. The other tool we use a lot, we use mouse models a lot now to model genetic disease because of this CRISPR stuff you've heard about. CRISPR is a bacterial enzyme that you can inject it into a cell, and it will precisely target a specific DNA sequence so that you can replace it with however you want. So when I started out in genetics, when I started my lab 20 some years ago, it cost about maybe $5,000 to $10,000 to make a mouse model, and it took 2 years. Today, I can put in an email and send it to the people that we work with at National Jewish here in town. It's called the Transgenic Mouse Lab. I can send them an email and say, "This is the mutation I want in my mouse," and they will send me the mouse in about 2 months, and it's probably $1,000. It's ridiculous how easy it is to use. We do the same thing with zebrafish, these little guppies that we have tanks. We have in labs with hundreds of tanks of guppies that you can manipulate their genome so easily. And zebrafish are nice because when they're developing, they're see-through. So you can take a developing embryo of a zebrafish and put it under a microscope, and you can see you can see every cell in the body as it's developing. We also use roundworms. They're called C. elegans. And the beautiful thing about C. elegans is that I think they have 960 cells in their body. The adult has 960 cells, and we know every single cell, where it comes from and what genes are turned on and off in that cell in development. So the tools that are available to us today are amazing, but they're all because of the people who came before us. Genetics, like every other field, we all stand on the shoulders of the people who came before us. And it's great that our children will be able to stand on our shoulders and do even better.
Dr. McCandless, I want to thank you again so very much. This was very, very interesting. And I know that we'll get a good turnout on our YouTube or Facebook, wherever it's being posted, because we always do. People can't quite make it tonight. But this was absolutely enlightening, fascinating, and second time I've heard you, and both times have been just fantastic. Thank you very much.
Thank you. Thank you very much for
Thank
the kind
you.
invitation.